So let's get started on the first session of this morning, or session 1-1, recent findings on cell atlases, continuing very naturally from what you've heard so far. And our first speaker here, and I'll be chairing the session with Sharmila Sengupta. Our first speaker is none other than John Randell, who's Chief Alliance Officer, Human Cell Atlas, talking about HCA data ecosystems. John, great to have you here.
Hello, everyone. Welcome. As John mentioned, my name is John Randell. I'm HCA's Chief Alliance Officer. I work in the HCA Executive Office. The Executive Office consists of approximately a dozen professional staff who are based in 4 locations around the world. As it happens, we actually have representatives from each office here today. My colleague Yoshi Ando from Japan, Lucia Robeson from the UK, Esther Chua from Singapore, who seems to be everywhere all at once. I'm not sure where she is at the moment. And then myself from the US office, which is based at the Broad Institute in the US. What I'd like to do today is really provide a little bit more of a window into the projects and data platforms that make up what we call the HCA data ecosystem. And I'll go through through this in 4 parts. The first being a little bit more detail into how these atlases are actually being assembled, and then I'll describe the components of the data ecosystem and how they all collectively support atlas assembly. And then I'll provide a little bit more detail into 2 developments that we're particularly excited about with HCA, one being the new data portal, which Aviv mentioned earlier, and then finally an update on a topic that I know is of interest to many of you, which is managed access or controlled access, and how that will play out on the HCA data portal. So you've already heard a lot from Aviv in particular about the process of atlas assembly. Just to review, each of the 18 HCA biological networks will contribute 1 or more draft atlases to the first human cell atlas. And this is, again, as you've heard, very much a collective process that anybody can be involved with. It is, as every element of HCA's science is, overseen by the HCA Organizing Committee, which is an independent body of roughly 3 dozen scientists, again, from around the world. It involves the HCA biological networks, of course, and they're supported by a roughly dozen-person team of computational biologists, which is really the integration core team, which works with each biological network to carry out the data integration that leads to atlas assembly. And of course, all of this is supported by all of you in the HCA community. And I particularly also just want to thank the Chan Zuckerberg Initiative, which is providing very generous support, particularly for this integration core team, which is spread across Singapore, the UK, and Germany. This is a quite simplified diagram for the many steps that contribute to the building of atlases. Roughly speaking, you can think about this as occurring in 3 phases. The first being the collection, the kind of harmonization of data and metadata. The second being the technical aspects of data set integration. And then third, the biological insights, which involves both cell type annotation of the integrated atlas, and as well as the sort of downstream biological discovery, which of course is the ultimate goal of everything that we do. Just to highlight a couple of key attributes of this chart, data set selection is done by the biological networks themselves. And so you've heard that you can contribute to the selection of data sets that go into the atlases by joining one of the biological networks. And to give you an idea of the scale, the biological networks are selecting anywhere from, say, 10 to 40 or more data sets to be combined into these atlases. So it's really quite a large-scale process. And even the first draft of the Human Cell Atlas will involve something on the order of 400 data sets, again, from around the world. The data sets are then, of course, subjected to quality control, batch analysis. There's benchmarking of integration approaches. And then the community cell annotation, which is a really critical component of HCA. And you'll hear much more about that later in this session from Evan Biederstedt. And just to highlight that each one of these steps needs to involve the biological networks. And so this is a process that's coordinated through the executive office, but really is spearheaded by the coordinators of the biological networks themselves in combination with the integration team. The process is, of course, iterative. We've learned a lot from the first wave of atlas assembly, and that's being applied to successive waves, and it's adaptive as well, which you would expect. So, again, we're learning as we go. We've learned a lot already and are applying those lessons to the creation of more efficient atlases going forward. You've heard from Vive that assembly is occurring in waves, and so the first wave of Atlas assembly involves the first 6 biological networks, 3 of which actually have been published. You heard about nervous system and lung, and I'll come back to that later. In addition, the eye network has a preprint describing the first eye atlas, which actually focuses on the retina itself, and that has been submitted for publication as part of the publication package that Aviv described. You also heard that the second wave is just getting underway right now, involving 6 more of the biological networks. I would particularly highlight the genetic diversity network, which Shyam just described. The genetic diversity network is actually one of 3 sort of cross-cutting networks, if I can call them that, the other 2 being organoids and development that really involve more than one organ or tissue or biological system. And as you heard from Aviv, but I think it's really worth emphasizing, what we're creating currently is only the first draft of the atlas. And so there will be shortfalls in anatomical coverage, in cell type identification, and so forth. And so that will require the integration of more transcriptomic data going forward. But as Aviv said, we also, going forward, will be integrating multi-omics data, spatial transcriptomics. And as you've heard in a very nice presentation from Shyam just now, a really important principle of HCA is that the ultimate atlas be globally representative. And so this is a major focus of our work in the years ahead, is to really ensure that we are capturing all elements of human diversity. Not just genetic diversity, by the way, but also geography and experience, which can also influence transcriptomics. And so as you've heard again, and you've already seen this slide, but I'll just show it again, you can get involved by joining the biological networks. I'll put this slide up at the end as well in case you can't capture the QR code, but that will lead you to a form where you can sign up for 1 or more of the networks that are of interest to you. Because it came up in the previous session in a question, I also want to emphasize that anybody can contribute a publication to the Human Cell Atlas. And if you go to the Human Cell Atlas website, there's a publication tab that has instructions for how to submit a publication to the HCA Publications Committee to be acknowledged as a contribution to HCA. And so I encourage you all to do that as well. I'll move now to, again, what we call the data ecosystem, which involves multiple independent projects and components, all of which collectively work to support Atlas assembly. You've heard about many of these already. They include the HCA data repository, which is here represented by the public-facing UI, which we call the Data Explorer, the cell annotation platform, which Evan will describe in his talk later, the Human Cell Atlas data portal, and I'll give you a tour of this shortly. And I'll also mention the Chen Zuckerberg Cell by Gene tool, which is an analysis and visualization portal that is useful broadly for single cell data, but specifically for HCA data, and will have a very specific role to play in HCA as well. Actually, before I go to this slide, I do want to say that I get a lot of questions about where to submit data, a lot of, I guess, what I would call confusion about the difference between submitting data to HCA and to something like Cell by Gene. Do you need to submit your data everywhere or just to one? And so these questions come up repeatedly over and over. They came up at the HCA Asia Meeting in Bangkok last year. So we've really put a lot of thought this year into how these platforms can really collaborate more closely to really support HCA. And what we're really trying to do is leverage the strengths of each platform so that collectively these ecosystem elements will really become greater than the sum of their parts. And so to describe how this will work going forward, and this is a very high level overview, but you'll hear much more about this in the months ahead. The HCA data repository, or again, this is the portal being called the Data Explorer, is going to be the home for raw data and rich biological metadata. By rich biological metadata, I really mean a broad set of metadata fields ranging from diversity to disease to other attributes of the donor and the patient. And part of the point of highlighting this is that in many cases, both the raw data and much of the metadata need to be under controlled access. And I know that this is the case for many of you. And so managed access is being built out through the HCA repository, and in the months ahead will come online and will be available to you to submit your data that does need to be under access controls. Conversely, Cell by Gene is open access, and that will hold HCA count matrices as well as sort of high-level technical metadata, and particularly the technical metadata that is important for the process of atlas assembly. And so this is really what we call kind of the matrix store for HCA, the main source of truth for what matrices are being used to build atlases. Again, you'll hear about CAP, or the Cell Annotation Platform, shortly, which is a platform for cell type annotation, a very important contributor to HCA, and then the data portal, which I'll describe now. But I also want to mention that in addition to HCA's data portal, we also work with other single-cell data portals, such as the Single-Cell Expression Atlas or the single-cell portal from the Broad Institute. We're in active discussion with them right now about how HCA atlases can be served through those portals as well. And that really sort of adds to the richness of tools that are available to the scientific community to explore HCA data. So I'll talk a lot about the HCA data portal in a minute, but really the idea is to have an ecosystem not just of these data platforms, but also an ecosystem of data portals. Okay, I'll move on to something Aviv mentioned, which is a particularly exciting development. 2 weeks ago, we released a new version of the HCA data portal. We sort of colloquially behind the scenes call this HCA Data Portal 2.0. I'm actually using that terminology here, partly to really emphasize that there will be a Data Portal 3.0, and there will be a Data Portal 4.0. And so as the Atlas expands, as new data types are brought on board, we also need to be sort of iterating on the data portal itself, constantly progressing towards that goal of having a single searchable human body atlas that is navigable and can be zoomed in on to identify information on cell types, their properties, and how they interact. But right now we're just in the second iteration, which was published 2 weeks ago, and this is available at data.humancellatlas.org. So I'll give you a brief tour of this data portal. This is the new homepage for the data portal. As you can see, currently in the HCA data repository, there is information on approximately 50 million cells from over 400 projects. There are two buttons here I'll highlight. One is what is the HCA Data Explorer, and the other is a button to contribute data, and that takes you to instructions for how to contribute your data to HCA. The HCA Data Explorer has itself been redesigned to improve navigability and sort of to make it easier for you to find the HCA data sets that are in the HCA data repository. So each one of these links takes you to that data set where you can download, again, raw data, but particularly raw data and associated information. But the new feature of Data Portal 2.0 is to organize information around the concept of biological network atlases. So on the bottom half of the screen, you see icons for each of the 18 biological networks. And next to the lung and nervous system icons, you see, you can't probably read this, but a number in a circle here. In this case, 1, in this case, 2. And that refers to the number of atlases that have been released by that, or published by that biological network. So you can tell that we're really in the initial stages of assembling the first draft atlases, but as atlases are published, they will come online on this data portal. So we're working on the Retina atlas right now, and then be accessible through the data portal. And the way I think about the HCA data portal is really as a gateway to information about atlases. And so you will see that there's information on sort of the scope of the atlas, who contributes, there will be links to publications, to code, and so forth, but then there will be links out to the analysis portals where you can actually explore the atlases themselves, as well as to the data sets that make up the atlas. And to show you what this looks like, this is an example of what happens if you click on the lung icon. You bring up the lung network's homepage, and so you can see information, for example, on who the coordinators are, and then at the bottom you can see the list of atlases that have been published by that network. If you click on that atlas name, that brings you to the Atlas webpage. This is the page for the first lung cell atlas, which was published in Nature Medicine in early June of this year. There's information about sort of a description of the atlas. In this case, they published both a core atlas and a larger sort of full atlas that included disease-oriented data sets. And so there's a sort of an explanation of that division. And then down at the bottom, there are links to where you can view these component atlases, like the core and the full atlas, in this case on Cell by Gene. And as these atlases come online on other portals, we'll also include links out to those other portals here as well. But currently, Cell by Gene is the main path for exploring these atlases. So you can link directly to that. We're actually very soon going to also be adding a direct download link. So we're working with Cell by Gene on that. So you can actually download the atlases directly from Cell by Gene through the HCA web portal. And then at the top of this page, I should also say there's a tab for source data sets. And so that's shown here. It just is a list of all, in this case, 49 data sets that went into the Lung V1. And again, with links at the moment to the data sets in the HCA data repository, and then eventually you'll also be able to link out to those data sets on other portals as well. I'll also show you the nervous system networks page on the current data portal, just because this is a nice illustration of what happens when you start publishing more than one atlas. And so at the bottom, you see that they have published recently in Science magazine, there was a publication package from the NIH Brain Initiative that included 2 atlas papers that we are considering first drafts of HCA brain atlases, one being a sort of whole brain atlas and the other being a deeper dive into the cortex. So that's the first cortex atlas. There's a lot of flexibility in how we do this, and so we work closely with each of the biological networks to sort of design their page and to incorporate special elements into the HCA data portal. The nervous system is also a special, particularly brain is a special case, but it nicely shows how HCA can work with other consortium, in this case, the NIH Brain Initiative, and specifically here, the Brain Initiative Cell Census Network, to sort of collect the best atlases and the best information and sort of incorporate them into the emerging human cell atlas. So that's an overview of the data portal. I encourage you to check it out. This is a constantly evolving tool, and so I'm sure we'll be rolling out many more features in the months ahead, but we are very excited about this. It's really the first time that we've been able to sort of publicly present HCA work in the context of individual tissue atlases. So next I want to move to the topic of managed access. This did come up last year. I know it's on many of your minds, and so I'm very excited to be able to give you an update today. This has been a project that has been in development for quite some time. So actually, before I move to the timeline, there are multiple instances where managed access might be important. One of them, of course, is with hospital data. In many cases, there are stipulations from IRBs or perhaps national regulations pertaining to data protection that require access controls or other sort of criteria placed on the downstream users of your data. And so that's the goal here. The challenge, as you heard from Aviv, with members in 97 countries, we could literally get data from anywhere in the world. And so we have to deal with many different regulatory environments and many sort of IRB policies in doing this. And so our goal here has been to put in place a system that we believe will cover most situations regardless of the provenance of the data. And so this could be GDPR in the EU, it could be patient data from hospitals in this region or elsewhere. You could also imagine that access controls would be important for particularly sensitive populations, which could be pediatrics, it could be indigenous populations, and so forth. So there's a wide range of use cases here, and we're trying to essentially get a one-size-fits-all approach, but also acknowledging that there will have to be some specific elements rolled out as we move ahead. Very briefly, we've been building this up over the year in terms of the infrastructure. We're piloting this with a handful of data sets right now, including data sets from the Asia Immune Diversity Atlas project. We are aiming for a full launch in the spring, and then maintenance and expansion will occur, of course, on an ongoing basis. And I want to briefly tell you about how this will work in the context of HCA. What you see here is actually how data flows in and out of the HCA data repository under the current open access system. And this is the data that's available through the HCA data portal. The data repository is a joint project of 3 institutions, EBI, the Broad Institute, and the University of California, Santa Cruz. And so as you contribute data, EBI with help from UCSC is responsible for data ingestion. The Broad Institute then stores the data in the HCA repository, and UCSC oversees the data browser. And then those data can be directly downloaded in an open access manner from the HCA website. There are numerous legal contracts that have to be put in place, though, for managed access to ensure that downstream users are bound to the restrictions on the use of the data. And so this is mediated by a nonprofit entity, which is known as HCA Incorporated, HCA Inc., which serves as the data controller for HCA managed access data. And in this system, you will actually contribute the data as before to the same data repository, but with the additional, shown in blue arrows, the additional requirement of submitting or signing an agreement with HCA, whether you're a contributor or a user of the data, and that will sort of bind you to the data protection requirements of these managed access data sets. You can be assured that the operators of the data repository will also behave responsibly because in yellow, we are executing agreements with each of those 3 entities that will also restrict how they can handle the data. And then finally, in addition to signing a data access agreement with HCA, there is already in existence an HCA Data Access Committee, and this will serve as the single approver of data access requests for managed access data. And this will happen through the data browser, so you'll submit your access request, it'll be approved by the DAC, and then the data browser will be instructed to give you specifically access to that data set. So that was a very quick overview of managed access. I'd be very happy to answer any questions you might have about that process, but just to summarize my talk, we've already been publishing some initial atlases, many others are in development. We have the new data portal, which is live and quite exciting for us. There are many different platforms that interact to form the data ecosystem, and we're really working on sort of improving the coordination there. It's quite dramatic. And then we are planning to roll out managed access in the spring. And I would just thank the funders of much of this work, the Chan Zuckerberg Initiative, which funds much of the work behind the HCA data platforms, Schmidt Futures, which funds CAP, which again you'll hear about shortly, and then funding from the Chan Zuckerberg Initiative, the Klarman Family Foundation, and Welcome for operational support for HCA. So thank you very much, and I'll end there.
Thanks very much for that overview, John. Fantastic oversight into all the activities. Questions?
Hi, wonderful talk. I'm Joveria from IIT Delhi. I'm interested in knowing how can we add spatial transcriptomics data to the atlas? Like how can we add an additional layer of spatial information to the existing?
Sorry, there's an echo up here, so I'm finding it a little hard to. You were asking about patient data?
No, I'm asking how can we add an additional layer of spatial information? For instance, I'm interested in knowing how can I add spatial data to the atlas? How can I contribute to that? To the existing, for example, lung cell atlas? Spatial transcriptomics data.
Yeah, this echo is. Oh, spatial data, sorry, yes. Apologize, it really is an echo up here. This is very much a discussion in progress. So you can submit, I know that the data repository does have some spatial data right now, and I would encourage you to follow that contribute button because that will take you to contact information for the HCA data wranglers who can have that discussion with you about uploading spatial data. I think in the sort of larger sense, this is really a major project for us for the next 2 to 3 years, is to figure out how to best both ingest those data, but also in particular to portray spatial data on the HCA data platform or the data portal itself. So that's kind of a stay tuned, but right now you can already talk to the data wranglers about what they're able to do in terms of ingesting your data.
Yeah, okay, thank you.
Sure, thanks.
Lucia, any questions online? Yeah. I'll just ask a quick question, like kind of a Cliff Notes version. If people want to contribute data, whom do they contact? Or where do they go? If people want to join an organ atlas, whom do they contact? If people want to download data, where do they go?
Sure, yeah, thank you. For contributing data, you can reach out again to the data wranglers, primarily based at EBI, but also at UCSC. That contribute button actually takes you to a full web page, which has instructions for how to do that. We're actually in the process of revising those instructions, but you can certainly follow there's an email address on the contribute web page on the data portal, which you can use if you're interested in contributing data. In terms of accessing the data, again, the data portal, if you navigate through the atlases, but particularly through the Data Explorer, you can search on data sets that might be of interest and download the data. And much HCA data is also on Cell by Gene and other portals, so you can also find it there. And then your third question was?
If they want to join an atlas. If they want to join an organ atlas.
Yes. There is information. Actually, if you go to the main HCA website, if you scroll to the bottom, in the footer of the website, there is a Join HCA button. That's the best way to join HCA. If you're not an HCA member, please sign up. But that process also queries which biological networks you might want to join as well. And so you can also use that Join HCA button to join biological networks.
Thank you so much, John. Next speaker is Oni Basu from University of Chicago.
Hi, everyone. I'm very happy to be here. I come from the University of Chicago. I'm going to be telling you a little bit about the Human Cell Atlas efforts that have been ongoing in my lab, and also I'm representing the University of Chicago in this. This is a work in collaboration with many of our colleagues who are at the University of Chicago and elsewhere. But I also wanted to say that, I am from Kolkata. The HCA Asia is very close to my heart, and I would like to find out ways to contribute. Partly also because my research interest happens to be in the direction of microbiome and infectious diseases. So without further ado, I would like to show you where the University of Chicago is. That's here. This is where the hospital is. I am in the hospital, UC Hospital, and I work with a team of clinicians and surgeons to get samples, get them processed very quickly. And to start off, the core technology that we use in the lab is droplet-based. So these are microfluidic droplets. Along with DNA barcoding that I had co-developed when I was in Avid's group as a postdoc, and since then I have set these up in my group, and we use this as very targeted approaches to do single-cell sequencing for. So here are two technologies that we use very commonly. One is based on single cells, and one is based on single nuclei. They are very interchangeable, and we tune them as required in the lab. So the first thing that we did when I started my lab was to compare single cell and single nuclei RNA sequencing. So DROP-seq would be single cell, DROM-seq would be single nuclei, and we applied it to human iPSC-derived cardiomyocytes. So we took iPSC cells from humans, we differentiated them over a period of 15 days, and we sampled them with both single cell and single nuclei technologies. So what we see here, UMAPs of both techniques. Both these technologies can resolve the cell types in this differentiation process. It can also resolve the cell proportions pretty well, and it can resolve the differentiation process shown by these pseudotime trajectories. Some other lessons that we learned is that not all cells differentiate at the same rate. Some cells are fast responders, some cells take their time to differentiate into cardiomyocytes, and that not all cells do differentiate into cardiomyocytes. So they do go into some alternative lineages, like shown here in alternative lineage 1 and 2. So these are in vitro systems. One might ask what meaning they might have in vivo context, and I think it has a lot to do with cell signaling, what it sees in the tissue microenvironment. Next comes the question of Human Cell Atlas. We have been involved in 3 different tissue types: the heart, the female reproductive system, and the gut. You might ask why these systems? The heart is relatively fewer cell types. It does one job, it's mission critical, and it is to pump. The female reproductive system starts getting more complicated. It supports one of the most complex functions in the human body, and then in the gut, more on that later. So we use single cell and single nuclei RNA sequencing interchangeably. We also add single cell ATAC-seq, which is an assay on chromatin accessibility at the single cell level. We then do integrative analysis, and for validation, we do immunohistochemistry, RNA FISH, and some spatial transcriptomics. So our heart and female reproductive system projects are supported by the Chan Zuckerberg Initiative, and the gut atlases are supported by the Hemsley Foundation. So we do use our droplet technologies, but we also are very avid users of the 10x Chromium platform, and this allows us to do not just the RNA part, but also the chromatin accessibility part. There's a lot of echo. So the first one that I'm going to be telling you about is the Heart Cell Atlas. We did single nuclei RNA-seq. This is a technique that is not amenable to single cell sequencing, so single nuclei was the only option. As you can see, there are not a huge number of cell types, and that will get clearer as we go on. Here's a single cell ATAC-seq, which is looking at the chromatin accessibility, and we group cell types by the accessibility in their chromatin, open chromatin regions. For example, here are two genes that shows both expression levels and motif accessibility, so transcription factors that are in the open chromatin regions, and they match very well. We are also interested in looking at open chromatin regions in cells and comparing these open chromatin regions. Are they cell type specific or are they shared between cell types? We are also looking at connecting this to GWAS studies to look at GWAS genes, and if we can do a priority scoring to these GWAS genes, which is a big collaboration with Sebastian Pott and Shin Hye, who are human geneticists. Okay. The next topic that I'm going to be talking about is the fallopian tube. We are working on a few different organs from the female reproductive system. Today I'm going to be focusing only on the fallopian tube. And we are also, in this part, looking at postmenopausal women. This is because a postmenopausal system is a much more well-defined system. And even so, there are many more cell types that you can see. Cell types, so ST are stroma cells. There are many different types of stroma cells. T and NK cells, there are subdivisions, endothelial cells, and so on. So our study in postmenopausal fallopian tube was also motivated by our other interest in ovarian cancer, where these two cell types, the SE and CE cells, ciliated endothelial and secretory endothelial cells, that seem to play a big role. Here's a dot plot showing the marker genes for each of these cell types, and as you can tell, they are these are marker genes, so they are uniquely expressed in different cell types. Here are variations in cell types that are caught between donors, and then we go back and do immunohistochemistry or immunofluorescence to match the numbers from single-cell RNA-seq to what is found in tissue. It's taking a little bit of time to respond. So as I said, we are interested in the ciliated and secretory epithelial cells. Here are some marker genes for those two cell types, and we go and look at different regions of the fallopian tube and see if they are indeed expressed. One thing that was very interesting to us was finding expression of many hormone receptor genes. The plots are not showing up very well. I apologize for that. But it was interesting to us because these are postmenopausal women where we expected there not to be a lot of hormone activity, but we do see indeed hormone receptor activities. And we went and verified that these are estrogen and progesterone hormones that are indeed present in postmenopausal women. Now, we look at different regions of the fallopian tube separately because they have different disease biology associated with them. Here's the fimbria, which is the closest to the ovary, and the ampulla and the isthmus. We do see region-specific differences between the different regions. We then look at single-cell ATAC-seq, and here are some ATAC-seq cell types that are found in each of these different regions, which are then put together to give this overall lay of the land for the ATAC-seq data. So for ATAC-seq, we usually do unsupervised clustering and the label transfer from single-cell RNA-seq. Here we see that in the isthmus and ampulla, for example, they have very similar chromatin accessibility patterns. Each of these columns are a different cell type, but the fimbria has a very different chromatin accessibility pattern for postmenopausal women. We then went ahead and started looking at premenopausal women. This is, we took the postmenopausal women as our ground truth and started integrating premenopausal women here. Now we see that looking at the SE and CE cells, the ciliated and the secretory epithelial cells, there is a larger variety of cells. For the immune cells also, there is a larger variety of these cells. And indeed, there are much higher presence of immune cells, much higher presence of secretory and ciliated epithelial cells in the premenopausal women, as opposed to postmenopausal women, where there are a lot more endothelial cells and vascular smooth muscle cells, which is something that we don't understand as yet. But these are just the cell type proportions. We also look at the differentially expressed genes. We look at the differentially expressed genes between the pre and the postmenopausal cohort, and indeed we find that they are very distinct in the fallopian tube. We are interested in this, as I said, these secretory epithelial cells in the pre and the postmenopausal women. We look for these particular type of cells called PEG cells that are associated with ovarian cancer, and also look at the hormone receptor genes that are associated with these cell types. And the pattern for the pre and the postmenopausal women are quite different. Another interesting thing that we found was the role of the stroma. We always think of stroma as supportive tissue, but indeed we see that the stroma is also responding in lockstep with the epithelial cells or the hormone secreting cells. And there are very distinct patterns that we see for the pre and the postmenopausal women. The menstrual cycle effect. So the previous slide that I showed you was the difference between pre and post. Now we are going to look at the differences between the two different cycles of the menstrual cycle. One is the proliferative, one is the secretory. Here's our data cohort that have a wide variety of age and BMI differences that can cause hormonal effects and so on. So here again, we see differences between the cell types between the two cycles, two phases of the menstrual cycle. There are differentially expressed genes that are different between the two menstrual cycles. OVGP1 is a gene that is associated with ovarian cancer, and they are differentially expressed in the two phases of the menstrual cycle. Again, we can associate them with the stroma cells. So there is we think there is a lot of cell-cell communication in the tissue microenvironment with the cells that were thought to be, the driving factors of biology, but the stroma is indeed supporting the biology. Here I'm showing you some ligand receptor interactions that take place between the secretory epithelial and the stroma cells. Another thing that we can do is to look at differentiation trajectories. So this is looking at the proliferative and the secretory cells, and we can see that these SE1 and SE2 cells are not really two different cell types, but two different cell states that are moving back and forth between the menstrual cycle. There was a question about the differences between single cell and single nuclei RNA-seq. The data that I showed you before was for single cell RNA-seq, but single nuclei RNA-seq is one of the things it's good at is to look at the ratio of pre-spliced and spliced RNAs and look at differentiation trajectories. And if you are, if you suspect there is a cell state type that you are interested in, single nuclei might be a good option. So we are also looking at not just gene expression, but also transcription factor activity across the chromatin cycle. So because these are very subtle changes, we had to do some normalization. This is pre and these are some of the motives, some of the genes that are present. We subtract the pre and the post relative accessibility, and then do a normalization to have these interesting transcription factors pop up. We then look at these keratin family genes, which are both associated with ovarian cancer, but also associated with the menstrual cycle, transcription activities, and transcription activity Z-scores. Again, a normalization pattern that, so these are very subtle differences, so you might require different analysis methods to have them show up. And here's the 10x Visium, images showing that these this keratin family genes, TUB and OVGP1, are indeed present in the fimbria region, which is the closest to the ovary. The last project that I'm going to be telling you about is the gut and Crohn's disease. Now, this project is a little bit different in that it has a very strong disease component. So we do look at pinch biopsies from healthy donors, but also pinch biopsies from Crohn's disease patients that are in remission or under active inflammation. We take sample regions from the small and the large intestine. These are very superficial epithelial biopsies, which means that we are not sampling very deep into the stroma. So that's a caveat. We then go ahead and do single-cell ATAC-seq and single-cell RNA-seq. We do the validation either by traditional methods and some spatial transcriptomics, and then we also establish some organoids, which I'm not going to be talking about today. So this project for the Human Cell Atlas is for Crohn's disease, but we are also looking at ulcerative colitis and a particular advanced disease stage called pouchitis and pouch. There's a preprint out for this, which also leverages some of the control data set that we generated for the intestines. So the small and the large intestines are structurally and functionally very different. You can think of them as two different organs, or you can think of them as a continuous organ. Their functions are different in that the small intestine is more associated with digestion and nutrient absorption, the large intestine with electrolyte and vitamin electrolyte absorption and vitamin production. The pHs are different. The microbes that are present are also different, which is a focus of my lab, but I'm not going to be talking about in this presentation. So here is indeed the picture. The cell type complexity gets really complicated. I am showing you three major cell types present, of which there is an immune cell part, there is a stromal part, and there is an epithelial part. The immune cell part and the stromal part are common to both regions, whereas in the epithelial part, they are specific to the large and the small intestine. So bottom, it starts from the stem cells, it goes into the colonocytes, it goes into the secretory lineage in the colon, and on the top, you start from the stem cell and you go into the enterocytes in the small intestine. So we do have region-specific and shared cell types in this picture. So here is again a very complicated picture, but really there really is a large variety of cell types present in the TI, the terminal ileum, and the ascending colon in the body. Another thing that we are doing, because we do have disease cases, we are looking at the number of interactions, number of DE genes that we can find from control regions, from samples that are not inflamed, samples that are adjacent to inflamed, and samples that are inflamed, actively inflamed. We can then draw cell-cell communication networks, and we do indeed find that as the inflammation status goes up, the number of interactions go up, kind of monotonically. If we look at the expression of genome-wide associated studies that are from IBD and Crohn's disease, we do see again that there is a higher expression of these GWAS genes, Crohn's disease GWAS genes, as inflammation status goes up. We are now applying some co-expression network and network analysis to the epithelial lineage alone, and we identify genes that are associated with this co-expression network, and drawing out the co-expression network. These are genes that are identified from the co-expression network analysis, some of them like MOOC2, MOOC1, and another one I cannot see. These are overlapping with the GWAS genes, but most of them are unique. And again, as inflammation status goes up, the network gets larger, more connected, more complicated. Next comes the question of validation. Yes, indeed, we are looking at control and inflamed samples, and looking at interesting cell types, quantifying if these interesting cell types are variable in the same way or in different ways between control and inflamed patients. Here I am, the green cells are marking TA cells, which are a sort of progenitor population, which seems to be increased in the disease case. And another case that I wanted to point out is a rare phenotype. So some of these patients do have a cell type that is not present in others. So, in disease case, you might find that, the number of cell types that are present, if you compare it to control, it's just, all bets are off, honestly. So we did find these cell types which we did not expect in the intestine. This is from the terminal ileum. They, in fact, are from the gastric glands. A part of this is a gastric gland metaplasia that happened in CD patients. So these are CD patients that are not inflamed. With that, I'm going to stop. Here are the people who are doing all this hard work. Lots of collaborations with clinicians and computational scientists and statisticians, core facilities, etc. Thank you.
Thanks very much, Oni. Questions? Lucia is up there.
I have another question from a member of the virtual audience, which Taba has asked, if we only collect or use single cell or single nucleus data, and if we don't consider post-transcriptional, post-translational modification processes as well as metabolism, how can we expect that to reflect the physiological function of the cell?
That's a very good question. I would say that we are not doing it because we don't have the technologies yet. We are using single cell transcriptomics because it scales well. If you want to capture the complexity of cell types, you would need to sample many cells. Many of the metabolomics or post-transcriptional modification studies, signal is very low, so you have to work with fewer cell types. So it is something that is important, but we don't have the technology yet, I would say. So probably a more nuanced, supervised approach needs to be taken, separate some cell types out, do the post-transcriptional modification assays or the metabolomics assays, overlay on the overall network. Yep.
So I have a question here at the back. So you mentioned you're using both a homebrew method of single-cell sequencing and 10x. Are there some recommendations as to when to use what? Because one is cheaper, one is a little bit more commercial one with better coverage.
We actually started using the homebrew methods and then 10x came into the picture, and then we adapted to the 10x technologies. These days we are doing mostly 10x for samples that work well when we are in the optimization stage, because there are we actually do a lot of tissue dissociation, cell type enrichment, dead cell removal. At that part we do use the homebrew method, which is cheaper, and once it works well, we then collect multiple patients, multiple regions, do 10x in that case. I would say that the computational methods that are out there for integrating different data sets, they work pretty well for both DropSeq, DrungSeq, and 10x platforms. Yeah.
Short question, short answer, Randell.
So there's great data on Colombia. So we are also interested in things like endometriosis. So do you have any plans, or do you have data generated on endometrium?
We do. So we do have endometrium data on, so in postmenopausal women, there is very little endometrium. We had a really hard time getting samples from postmenopausal women. We are focusing on perimenopausal women for the endometrium, and also premenopausal women. We do have multi-omics data for endometrium. We are still analyzing it, I would say. Yeah. Coming soon.
Hi.
Thanks. Sorry, we are out of time. Maybe more questions in the break.
Okay.
Thank you very much. I'm going to pass it to my co-chair to introduce the next few speakers.
Thank you, Shyam. The next speaker of this session is Jongil Kim, who is a principal investigator of the Seoul National University College of Medicine.
Hi everyone. My name is Jongil Kim from Seoul National University. I'm very happy to share recent updates for two single-cell atlas we are constructing. The first one is SCAID. It's mentioned by Chayam in the previous mention. The SCAID means the single-cell atlas of immune disease. It targets autoimmune and inflammatory diseases, and it contains many different sequencing platforms. So it's funded by Korean government, Ministry of Science and ICT. Initially, more than $15 million allocated for 6-year project. So we are targeting 16 diseases and more than 800 specimens and samples, and more than 10 million cells, and 24 different labs or PIs are involved in this project. This is a list of the 16 diseases. We selected these diseases by the six different criteria, and we hope to include at least 36 different donors or patients for each disease. For each disease, we will use solid tissue and or PBMC for the sequencing. And some diseases like rheumatoid arthritis, we will use synovial fluid, and lung disease, we will vial fluid for the sequencing. And many different sequencing platforms and spatial technology will be included. One of the uniqueness of our project is the city, Seoul. For the success of our project, we need two key strategies. One is that we should include a lot of large number of samples with various aspects. It cannot be done easily by a single hospital or center study. Another important thing is that for the reducing of the batch effect, using the sample transportation time, the sequencing or library construction will be done in small number of centers. As you may know, Seoul, more than about half of the Korean population live in or near Seoul, and the people living outside, who are living in outside Seoul, they usually visit so-called big 5 hospitals in Seoul when they have major illness. So we made 2 sequencing centers in Seoul, and all the samples easily transported to the sequencing center, and reducing the transportation time. And I think our project is well aligned with the second phase of the Human Cell Atlas, which aims to add more patient samples and increase the geographical and ethnic diversity. We started our project last year. Now we are still in the phase of the sample collection and sequencing, but we hope to open the first version of Atlas by the end of the next year. It will mainly focus on the single-cell RNA and some single-cell attack sequencing, but at the end of the 2027, we hope to present more comprehensive, complete version of our data atlas containing different type of sequencing platforms. The 3 students in our lab are actually contributing this project, doing all the administrative works as well as the science work, and among many different collaboration researchers, 2 key persons, Dr. Jongil Kim in Seoul University and Dr. Jung-Chau Lee in KAIST, are in charge of the sequencing and the library construction center. So for the PBMC sequencing and tissue sequencing, this is located in Seoul, north part of Seoul. This is southern part of the Seoul. This is the end of the first part of my slide. I anticipate that our project can extend beyond the initial intention. Well, for example, recently, the Korean researchers interested in asthma, which is not included our original plan. They contacted me and they wanted to integrate their data sequenced by different budget, different funding to our data for joint analysis. And many Korean BI researchers, which were not included in the original, the applicants, they joined and actively contribute their data analysis. And Siam and Jay in Singapore also contributed a lot by the member of the SAB. And I strongly hope to that our project can evolve into more international version, international initiative beyond Korea, including many different ethnic groups, especially from Asia. The second part is the OSCA Organoid Single Cell Atlas. Organoid is one major part of the HCA networks. And in the paper published in 2021, Dr. Bok mentioned that single-cell sequencing of organoid can contribute to validating or improving or some analysis of the perturbation data. But when we designed our project, our major motivation is the single-cell EQTL analysis. As you already know, the most important resources for the EQTL is GTX. But it's very useful resources, but it has two limitations. One is the imbalance in ethnic diversity. As you can see, only 1.3% of Asian samples are included in the GTX data set. So when we found some very interesting Asian-specific SNPs by GWAS study, and we want to check the EQTL. In many cases, we cannot find any data of the EQTL data from these databases. The second limitation is that it's mostly from the bulk sequencing, so we cannot find the contribution of the each cell types, especially the cell type is minor in that organ. When we designed our project in 2020, there is very limited publication about single-cell EQTL. Now we found a lot of papers published for single-cell EQTL, but you can see that most of the papers come from the PBMC or IPSC. This is because that single-cell EQTL analysis from solid organ is still challenging. I think this paper is the only paper comes from the solid organs. When we first designed our project, we intended two solid organs and lung and stomach. But when designing the project, we finally decided use the organoid rather than solid organs because of these reasons. The first one is the most important thing is best budget saving. So we absolutely needed full single-cell sequencing for the budget, but at that time we could not convince that the single-cell sequencing from the frozen sample was successful or not. So we decided to use organoid for the full sequencing. And by using the organoid, we could focus on epithelial cells in each organ. For example, when do single-cell sequencing from lung, we can find almost half of the cells from the immune cells. So if you are interested in immune cell, like a SCAID project, it's good. But if you are interested in epithelial cells, it's not that good for the efficiency. And we could reproduce the expression data if we want, and we can validate the EQTL result by some genome editing if needed. And we can find some EQTL, response EQTL to the stimulus like virus infection. And 2 months ago, we published a paper of the result of small-scale sequencing data. This is from the biobank. We selected 27 donors with different platforms, and we published this result in this paper. But I do not have the time to introduce this data. So please read this paper if you're interested. Now we are finalizing our data for single-cell EQTL for lung, and we used about 300 single-cell sequencing from 11 libraries. But finally, after QC filtering, we selected 146,000 cells and from 68 samples. And this is the analysis scheme. So we used genotypes by the microarray, and finally this is after all the filtering steps. We used the 9 cell types with 68 samples, and we compared 5 million variants with 14,000 genes. And finally, we could found more than 900 significant EQTLs from 500 genes. After that, we tried to co-localize with the long GWAS data, and we also did some the dynamic EQTL for the mixed model. So this is the scheme, the result of the EQTL result. So the most of EQTL were found in basal cell type of secreted cells because it's the major cells in our organized population. When you compare our EQTL data with the GTX lung or GTX multi-tissue, 70 or 90% of the EQTL was significant in GTX data. Interestingly, the 100 EQTL were not found in GTX lung, but found in other tissues with GTX. Probably because these cell types were minor in lung, but major in different cell type. And among these, most of them were matched in the direction of the expression regulation. I show just some examples of data. One is the example of E-station specific EQTL. This SNP is E-station specific, so it's very rare in other population. So it's significant only in basal cells, and it's located in promoter region. So we believe that it is the SNPs the important in the regulation of this the gene of the PLAUR is reported as associated with the long-lived phenotypes like this. Another example is the dynamic EQTL. Dynamic EQTL is that the EQTL found in the differentiation steps. As you can see, when the cells differentiate in this, from basal cell to ciliated cells, you can see these only in deuterosomal cells, these SNPs has significant EQTL. But interestingly, when we compare the single-cell text data from public one, this site is open in only deuterosomal status. So this region is open in specific stage of differentiation. And you can see the DNA H5 is an important gene in the ciliar function. So it may contribute to the function of the differentiated ciliar cells. Finally, this is an example of co-localization. When we compared the long GWAS data and EQTL, this sequence, this site is co-localized. There's the basal cell EQTL and asthma GWAS. And this gene, TSLP, is reported to associate with the asthma in many different previous studies. Interestingly, this SNP was not E-station specific, but was not reported in long GTX. Probably because it's significant only in basal cell, but in organ, the basal is the major one. But in tissue cell data, the basal cell contains very small portion. So that's maybe the reason that this EQTL was not found in long GTX tissue data. Finally, we are doing some validation study, but it's not easy to validate each SNPs. One simple strategy we used is the long-read RNA sequencing. So when there is some heterozygous SNP, we could find the heterozygous SNP by the genotyping, And if there is some early SNP in coding region, we can find the allele imbalance in the single-cell RNA sequence data. For example, we just used the 3 samples for long-read sequencing. This is the same site. So all the 3 individual has heterozygous SNP in this invariant region. But when you see these 2 samples, there is some quite imbalance in allele expression in these 2 data. And interestingly, this one has same distribution. That means that even though they, this patient, this sample has homozygous in the invariant, but because of the loss of allele, they are heterozygous in this coding sequence, and there is no disbalance. So that means that the site is the balanced expression region, but these 2 samples who has the heterozygous in the invariant has imbalance in the expression profile. So because of these only 3 samples, we are have some trouble with making these p-values of each site, but we managed to show some significance, the difference in the distribution by the statistical method. This is the member contribute this project. For the first part of the OSCA, long single-cell sequencing, Dr. Uchan Lee and Dr. Jongil Kim and Seon Lee, he is also active member in SCAID, did a great job. And for the equity analysis, Dr. Jae-Yong Choi and Jongil Kim is doing making the manuscript. And they also, these people contribute as collaborator to this project. Thank you for your attention.
Thank you, Jongil, for the excellent presentation, and we'll now take questions.
Hi, I was really fascinated by the work that you're doing with long-read sequencing and variants. And I noticed, are you considering the nature of transcript isoform diversity, which is generated between individuals, which is different between individuals when we're talking about presence or absence of specific variants? Because in your figure, it seemed like at the top,
Can you show this slide? Okay, you mean this one? Yes. Yeah. No. This is the sequencing allele frequency. So if there is G and C in this region, the variant, for example, is C. The frequency of C is 0.75 or 0.96, even though it's heterozygous in genotype. So that means that the expression of one allele is dominated than the others.
What kind of transcripts that they are making? What kind of transcripts? So you can actually have differences in the actual transcript which is getting expressed, complete transcript between the individuals. And then on top of that, you can have genetic diversity, ensuring that that particular exon is there.
We did not consider the different isoforms. So when we only found the exon was included in the sequence data, so we compared the various frequency with the 2 alleles. So the same site, but different alleles, the reference allele and variant alleles.
Any more questions?
Jay?
Yes, please.
Thank you for really fascinating to do. Is it IPS derived drops or IPS derived samples?
No, other stem cell derived, so tissue derived stem cells.
So I'm curious how you can maintain the proportions of different colors so we culture these cells. I suppose we culture recorded.
No, so we maintain any different culture by one, When we only sequence, when you do sequence, we pull the samples. Thank you. Thank you.
I don't see anybody else raising their hands, so is there anybody over there raising hand? No, I don't think so. So shall we move on to the next presentation after thanking Jongil? Our next speaker is Chung-Chau Hon, who is the team leader of RIKEN. And this is going to be a virtual presentation.
All right, good afternoon, everybody. It's morning there, it's afternoon there. Yeah, good afternoon, everybody. I'm currently in Japan now. Sorry, couldn't make it this time, but I got some exciting stuff to share with you guys. So today I'm going to talk about some work on atlasing the transcribed dysregulatory elements as single-cell resolution. All right. So that everything that you see today in this talk is from this bioarchive that we posted maybe 2 weeks ago. I highlighted some of the individuals in the study, key people. They are all basically in India now, except myself. But you know, go talk to them if you have questions of what I'm saying in this talk, and in particular, Jonathan, that with having a poster in the poster session, I guess. So let's move on. I'll start with some very basic introductions on why we're studying transregulatory elements. And we knew that disease predisposition and dysregulatory elements are highly correlated. They're linked to each other. So the genetic variations in which is highly enriched in dysregulatory elements, and they are one way or the other may affect gene expression, and you know, contributing to the disease predisposition among various diseases. So and our job is to look into the activity of these dysregulatory elements, in particular in those cell types that are physiologically relevant to particular diseases, and assess how much does those cell types contribute to different diseases. So talking about dysregulatory elements, here that we are particularly talking about those that are transcribing. So in particular, if we want to map those transcribed dysregulatory elements that previously that we have developed, different methods on the bulk level to capture the 5' ends of the RNAs, which in turns that you could use those information to look into the transcribed dysregulatory elements. That if you're interested in back a few years ago, that we've done a collection of analysis on the bulk samples, which is in the Phantom 5 project. It's a couple of years ago, but this stuff never gets old. So if you're interested, check a little bit via the QR code. There are 15, 20 papers in there. So, and of course, like moving away from the bulk analysis, actually a couple of years ago, that I think Piero and Jay also in India now, that Jay, Piero, Jonathan published a paper that using basically a C1 cage, a C1 platform to capture the 5' ends of the RNAs and mapped, the enhancers, showing the enhancers the RNA is actually detectable, even using a single-cell resolution. So, and moving away from the C1 cage platform, we actually also tested, different protocols within the 10x platform to see that how much does the enhancer RNA could be recapitulated with different protocols. In particular, that the 10x protocol is using oligo-DT priming, and we intentionally switching the oligo-DT with the random primer to see whether there is an enhanced detection of RNAs. But the result is actually surprising that we found that the oligo-DT priming actually are detecting the eRNAs as good as the random priming out of our expectation. And we think potentially this is because of the internal priming of the oligo-DT in the middle of the RNA transcripts. So what you're seeing here on the screen is the typical bidirectional transcribed patterns at some of the cage-defined enhancer RNA locus, and you can see both protocols detect pretty well at similar intensity. And then we decided, to move on with the original 10x oligo-DD priming protocol. So, the protocol itself is original from the 10x protocol, but cleaning up the data is actually a little bit complicated. So we wrote softwares to extract the true TSSs from the 10x data to distinguish those TSS from potential noise. That our original software was released a year ago, and a couple of months ago that we come up with a more improvised version of it. Both softwares are based on logistic regression of the TSS cluster properties, but the Camel TSS package actually also incorporated some deep learning on the sequence features as one of the features for logistic regression in collaboration with Yuan Hua from Hong Kong EU. I'm not going to get into details of that, but the consequences with the software plus the original 10x protocol, that you'll be able to detect the transcribed cis-regulatory elements in any regular 10x 5' oriented protocols. So, and then we move on and apply the 10x original protocol to 23 different tissues. And we defined tCRE using the softwares that I mentioned in about 340,000 cells from 23 tissues, and we defined about 180K transcribed cis-regulatory elements. So this is an effort that is from, most of the samples were from Japan. This is, we call the single cell medical network, involved multiple universities and hospitals in Japan. This is what the atlas look like. At the first glance, that you could see that we have around 21 distinct cell types that we could define, which is the obvious one that we call it level 1 cell types. They are distinct on the UMAP. That the numbers that you're seeing here, so these UMAP look a little bit different from the typical ones because we are overlaying the meta cells on top of the single cell. So you're seeing a little bit big dot and the small dot. The small dot at the back are single cells. This big dot in front are meta cells. So we are pulling something around like 50 to 100 cells into one meta cell. And most of the study, most of the data that we're using in this study is actually on meta cell level. So on the right-hand side, that you're seeing an example of how do we define the so-called level type cell types. Altogether, that we define about 180 level 2 cell types out of these 21 level 1 cell type you see on the left. On the right-hand side, that here you see that we take endothelial cell from blood vessels. For example, that you can see there is a clear distinction between capillaries, arteries, and veins. And also they are from different tissue origins. It also indicates that the integration of the data actually looks pretty well. So in this heat map showing right here, that you might appreciate there is many cell types like stromal cells, the fibroblasts, endothelial cells, they are coming from multiple different tissues. And of course, while the neural cells are only originated from the brain. So that we defined about 180K tCREs, and about half of them, we call them proximal tCREs, and those are the ones that basically overlapping with the gene promoters, like what you're seeing on the left right here. So this is one particular example that I'm showing you. These signals in promoter 1 and promoter 2, and they are differentially used among different level 1 cell types in here. On the right-hand side that you're seeing, on the contrary, are the distal transcribed cis-regulatory elements, and you could consider that most of them are possibly enhancers. That I'm showing you one example that these particular promoters linking to multiple enhancers, which I will show you how we link the enhancers a little bit later. So here that I could show you right here that the majority of the tCREs right here that we are sampling are one way or the other having some epigenome support from external data sets. So that means the CRE that we define are pretty valid. So we are linking these digital CREs to promoters by pulling multiple data set that I'm showing right here, including chromatin interaction data set as well as coactivity from the atlas itself. Here that we observed one particular interesting phenomenon, that while a promoter is more ubiquitous, which is measured with the Gini index right here on the y-axis, they are generally connected to more distal CREs. So like one particular example that we are showing here, this particular promoter is active across many different cells, in particular immune cells, but it's actually using multiple different enhancers across the immune cells. So here that that we conclude, and perhaps that the gene-specific regulation, cell-specific regulations of broadly expressed gene is actually supported by multiple distinct set of distal CREs across multiple cell types. So here that we also found there are a lot of long stretches of enhancers, which looks like super enhancers. That also means that tCRE could also detect super enhancers. And here that I'm showing you examples of a locus at the CD44. There's one particular locus we call that is high intensity frequently interacting locus. And some of these locus are unrestricted. Some of these locus, about 1/3 of these are unrestricted. It expresses across multiple cell types. Another 2/3 are very cell type specific. And when we look into the enrichment of heritability, in this case, we take Crohn's disease. We look at the enrichment of heritability, comparing between the promoters and the enhancers, or we say tCRE versus distal tCREs. In general, the distal tCREs are more enriched with the disease heritability. On the right-hand side that you're seeing here is a comparison between the heritability enrichment in the high-fi versus non-high-fi, that you can see there's a very clear enrichment across all the cell types. That means the high-fi that we defined is actually generally more enriched in disease heritability. So the next part of the talk is really about that, how we could take advantage of the single-cell resolution and look into the enriched tCRE usage at disease locus upon single-cell resolution. So the idea is to see that whether we could project the enrichment of heritability at single-cell resolution, so that we could assess the relevance of individual cells in disease heritability. So I try to keep it simple. So the major idea is that we look into the so-called tCRE modules, and you could consider, and those are similar to co-express modules, but it is more in the latent space. We use a CNMF. So the figure that you see on the left is the module activity across the 3,300 metacells. The figure you see on the right is this usage projected onto the UMAP. So you can see right here, these module usage are very, very specific to particular cell types. And right here that you're seeing the properties of the modules that we defined in a subset of cell types. We picked the stromal cells in particular. On the left-hand side, this is the usage across different level 2 cell types. You see those are in general very cell type specific, as you see in the UMAP before. And they also enrich in very different, very distinct set of gene sets, as you can see right here in this heat map. And they are also enriched in relevant disease heritability, if they are specific to certain cell types. So I'm highlighting here is the smooth muscle cells. You have 3 different subtypes, and there's 2 modules enriched in that. And this smooth muscle cells is also very enriched in heart attack heritability. So we actually took these heritability and projected into metacells. And here that you can see on the heat map on the left is the heritability of different diseases across the 3,300 meta cells. And you can see very specific association between cell types and diseases. As you can see right here, on the right-hand side is the projection of these numbers of heart attack to the UMAPs that I showed before. And we could use this information to quantify how much of every single cell type enriched in every diseases. So here, just showing you one example of myocardial infarction. So, and the data itself allow us to group the cells into different groups and easily to look for enrichment of heritability by something we call cell enrichment analysis. And right here that I'm showing you that if we run through the cell type and specific analysis, the fibroblast that is highly enriched in heart cells, in heart tissue, is highly enriched in the myocardial infarction, while the others is not. So it also shows you the power of the resolution that we have that could use to infer microenvironment by grouping the cell into different tissues. The other way of using the data is that we could take these closely related diseases and we project the score onto the UMAP, and we could make a differential disease score and look into what cells is enriched in this differential disease score. In this case, that we found the Crohn's disease are much more enriched in the fibroblast and stromal cells, and the ulcerative colitis is much more enriched in the immune cells. So let me go through the last two examples. And this particular ice cream score that we introduced would also be very useful in pinpointing relevant disease association of variants to genes. So particular example here is, so we are projecting the RA score onto the UMAP and this highly enriched T cell. And if we look into the correlation of the expression of the hi-fi locus versus the T cell within the T cell cluster, we found there's one particular hi-fi locus is highly correlated with ice cream score, while the gene that links to the hi-fi locus is not correlated with the ice cream score. So what it means, it might be more relevant to look into those disease with locus outside the coding space, which might actually give you something a little bit more specific than actually looking into the coding gene themselves. In this particular case, that the target gene of the locus is highly is broadly expressed versus the distal locus is very, very specifically expressed. So the last example that I'm showing you right here is linking the disease variants and alternative promoters. So one trait that we picked is body height, that we look into the enrichment of body height. Right here is highly enriched in the cluster of the stromal cells. And we are particularly interested to look into the chondrocyte because the chondrocyte is apparently related, to skeletal muscle, skeletal growth. That we are projecting the ice cream score of height within the chondrocyte cluster. Right here, you can see the little gradient across the cluster. If we take the genes that is correlated with this gradient, they are highly enriched in bone ossification, ECM growth factor, and SMAD. There's one particular interesting observation right here. There's one particular gene named WWP2, which both of the promoters is having height-associated variant, while only one of the promoter is correlated with the ice cream score, the other is not. And they produces two different isoforms. It's supposed to be having a very different effect in the endochondral ossification. So, and this is one example illustrating the value of looking into individual transcribed cis regulatory elements instead on the gene basis. That's all I want to talk about today, and I hope I convince you that looking into the transcribed cis regulatory element in a cell type specific manner in single cell resolution actually would provide very fresh and unseen insight compared to the traditional bulk and gene-based analysis. So acknowledgement and thank all the collaborators and the patients in generally donating the tissues so that the project made possible. And of course, the colleagues at IMS with Piero and Jay initiating the project, as well as Jonathan did all the data analysis. And that's it. We are hiring now, so if you're interested in a study, like Sushi Hot Spring single cell genetics, come and join us and send us an email. Thank you.
Thank you for your excellent presentation, but unfortunately we are unable to take questions because we are running late. I hand over this to my co-chair for the last talk.
Next we have Evan Biederstedt from CAP, which we've heard of already. Evan.
Thank you so much, Shyam. So we're out of time, and I'm between you and lunch, so I'll try to be quick. I'll try to be entertaining. Thank you for the organizers for inviting me. Thank you to Partha. Thank you to the Liver Foundation of West Bengal for getting me here. It was a bit of a challenge. I'd like to talk about CAP. I'm Evan Biederstedt. I'm at Harvard Medical School, the Broad. I also think I have an affiliation with Massachusetts General Hospital. So I'd like to talk about the cell annotation platform. I planned for a 10-minute talk, and I could try my best to even make it a bit shorter. There's three things I want to talk about. One is mostly just the motivation for why we're doing this project CAP, which is discussing the problem of cell annotations. That'll be the majority of the talk. Then there'll be a very quick just sort of overview of the software itself. There's been other talks I've given and YouTube videos on this subject if you want to look this up. And then I want to really engage with you about the cell annotation jamborees. You are the community with whom we should be speaking about how to best organize these. So I'd like to hear your views about this. All right, so why do the cell annotation platform? This is really, there's two big problems at the genesis of this project that we were trying to solve. One is we noticed there was basically a community standards issue with regards to cell annotations, how we share these and publish these within the field, which I'll get into. And then there's a larger problem here about what are the standards of cell annotations you need for creating human cell atlases. And, I think some people at this conference, you're starting out in single-cell genomics, so I'll give a brief overview of how this works in terms of cell annotations. This is looking at a paper from Peter Karchenko and Igor Adamienko at Igor's at the University of Vienna. Basically, you look at gene expression of UMAPs and of the clusters that you've clustered as at the end of your processing pipeline for single-cell genomics, and there's various ways that researchers look to see what are the identities and the functions of these cell types. You can look at gene expression, you can do various different types of differential expression, either using the background as sort of a baseline, doing differential expression between different clusters. At the end of the day, researchers will look to see sort of genes that will pop out at them that will show the identity of these cells. HBB is associated with hemoglobin production, so this has something to do with red blood cells. And since the advent of single-cell genomics, people have been making these UMAPs, which are colored and they're labeled. And there's been many talks today as well who have done this. This is great. But there's a couple of problems with this that really started this project CAP. One, just in terms of bioinformatics, even what we're doing now of simply giving these bioinformatic file formats like an AND data file or a SARAB file with labels, it's really not enough context or not enough information for you as a bioinformatics scientist to really do your job. It's not clear how these cell annotations were created, especially now in the days with algorithmic cell annotations. You've heard a bit about this with Aviv Regev, whereby there's different ways to do automated cell annotations, various different ways to do manual cell annotations. There's a basic question here about what do the labels mean. Even with the talks today, there's been different ways that people have worked with these labels. They could be abbreviations, they could be referring to different cell identities. Who did this? This is normally something associated with the papers, and then when we can get into with basically just the versioning of these cell annotations. And not just to pick on Peter, this is a paper by, I think, Dana Pear, Aviv Regev, and Charles Sawyers a couple years ago where they're looking at prostate tissue. And one of the main claims of the paper was that they said that there was 3 different types of luminal cells that they found within the prostate. You can imagine if you're a bioinformatician downloading public data, you'll have cells labeled luminal cell 1, luminal cell 2, luminal cell 3, and that's really all the context you have. So there's some basic transparency questions here and scientific reproducibility questions. And there's basic questions about how do we as a community communicate with each other about what do these cell annotations mean. You as a bioinformatician or as a research scientist, you should have some way to know whether you trust these cell annotations or you disagree with them. So that was the one motivation within this project, which I can detail for some solutions. Second is mostly just creating these human cell atlases. And that's really, you can imagine if you take two data sets from two research groups, you'll run into these sorts of issues. So you'll see that the cell annotations themselves, they'll be free text, they'll be used inconsistently, there'll be abbreviations. You can see this, kidney cells, immune cells, where there's abbreviations in some. Lunal 1, 2, and 3 is a pretty good example of this. There's different granularity as well, and this is mostly based on the publication. So on the left, they were interested in prostate, and so they have, they really annotate with very precise annotations, things like the seminal vesicle epithelium cells. And then on the right, because they're interested in adrenal gland, they had very precise annotations for the adrenal gland, but if they weren't interested in the study for like kidney cells or liver cells, they would just call them liver. This can't work with the HCA. We have entire bionetworks which are devoted to things like the liver cell atlas or the immune cell atlas. So these sorts of cell annotations simply won't scale for something like the HCA. And then there's maybe, I should just be a bit more vocal about some of the issues in this field. Biologists, I was thinking about calling the slide biologists have opinions, but biologists think, but researchers don't monolithically just agree on the definitions of these cell types. They don't. It doesn't work the way that you annotate your cells, and therefore that is the definition between all research groups, right? And I think what's happening now in the field is we have these large sets of data sets which have been curated within a certain degree of logic about how do you work with these cell annotations, and people are simply merging them together as if this is working with SNPs within the 1000 Genome Project. This simply is not the case. If you talk to biologists, we vigorously disagree on some very basic definitions of these cells. What are the marker genes you use to define these cells? What's the difference between the cell type and the cell state? And you can imagine just two situations whereby research groups are providing different labels to the same the same groups of cells with the same molecular signatures, or you can imagine a situation whereby there's two clusters with distinct molecular signatures between research groups, but they're given the same label. If you give them the same label and you associate it with, let's say, an ontology term and simply merge them together, it's not actually, you're actually propagating error when you do these sorts of things. And it only gets worse when you sort of input these into machine learning models, because machine learning models are only smart enough to learn what you've given to them. So I'm pretty convinced if I were to just take some of these cell annotations and randomly permute them and put them into machine learning models, many of our, I'm not sure the models themselves would notice a difference. Well, they would notice a difference, but I'm not sure if our colleagues would, to be a bit rude. So what's the solution? The solution is we need some community standards with how we're actually recording these cell annotations. So I don't have enough time to go through each of the definitions in detail, but researchers should be allowed to annotate cells with any term that they wish, and they can use abbreviations as well. We should ask for the rationale and the explanation of why they're providing this annotation the way that they are. What are the marker genes that they use to define that biological entity? And even questions like what are the canonical marker genes for this biological entity that they're referring to? Systematically collecting within the HCA things like the synonyms used to define those cells and various sort of category terms. The more broad cell labels for these cell types is something that, if you had this information, you would be able to do the analyses we want to do. And in terms of the first point with just basic bioinformatic analysis, the standard now is simply just to put these strings within these bioinformatic files. You have no context to know whether this was made by someone who is maybe studying this biological system for decades, right? They have expertise within this biological system. You would really want to trust their cell annotations versus a computational algorithm that was just quickly run, that has some known error, right? Without this context, you as the consumer of this data and you as the researcher can't really tell the difference between, how much you should want to trust the cell annotations or not. So we have some standards now within the HCA that we're asking all users to provide for the cell annotations. This is what we can use for building the atlases and, of course, for the upcoming cell annotation jamborees. So quick overview of CAP, at its heart, it was really just a large data repository, a way for us to create some sort of like GitHub-like workspaces for people to work on their cell annotations in private workspaces. publish them and share them with the community. We created fancy UIs, which I'll go through, which allow users to do these sorts of analyses on a web browser, calculate things like gene expression, search for specific genes, differential expression between the background and differential expression between different selections, clustered heat maps to see whether larger clusters should be broken up into smaller clusters. And if you collect this information, well, I'll get into this, there's publications. One reason we have to actually timestamp cell annotations is the data itself will be static, but the cell annotations will change. Even if you follow the logic of how we're building these atlases within the HCA, the more cells we aggregate, it could resolve some differences between cell types. So those annotations should be timestamped, so you can go back and see when we made the first atlases, these were the cell types, but after we gathered more data and after we corrected various batches and so forth, these were the cell annotations. And you, as the consumer, will actually be able to work with these in a way which I think makes sense. Running out of time here, we created these workspaces, which are like folders in terms of GitHub. There's various user roles you can use to work with these. This could be a long talk on its own. There's basically this interactive exploration UI you can work with at the UMap. You can zoom in, zoom out to do differential expression. We have a heat map that you can zoom in and out and look at the expression of individual genes. And as I said, if you systematically ask biologists for all of this context, you can start doing what was the original goal of CAP, which is to do this large crowdsourcing effort, right? If I am able to ask all HCA resources, what were the marker genes that you used for these annotations? What were the synonyms? What were the rationales? In sort of standardized metadata, I can actually create sort of summary databases, knowledge bases, explaining to the community, actually there are some disagreements about what are the marker genes you're using to find these cells, and what are the connections in terms of just the molecular data as well. And we can do this with all data sets that exist within the HCA. Last topic, and I'm out of time, is that there's this notion of the cell annotation jamborees. And I tend to think this is really the most valuable thing, well, I won't say the most, there's other great valuable things about the HCA, but speaking as a computational biologist, if the HCA didn't exist, we would have to invent it, right? At the end of the day, what we're doing is we're taking this integrated object, and we want to ask the research community what is their opinions of these cell annotations. We allow them to give as much rationale as they can, and basically collected this data such that we can figure out what is the consensus. I've told you that I can, just the input data sets themselves, I can provide some basic statistics of what is consensus. The consensus between, these are the shared molecular signatures of various clusters between data sets. This is the shared this is the shared metadata between the cell annotations. But it's really at the cell annotation jamborees whereby we put all the biologists in a room, experts from around the world, and we ask them to resolve some differences. And this is really the things I'd like to ask you about. This is me celebrating the world's largest democracy here. I think on the left was a local election in Karnataka, actually. But basically, there's some open questions about how do we best organize these cell annotation jamborees? How do we work with having agreements versus disagreements? Should we have several rounds of discussions, several rounds of voting? What does really consensus mean, even after you collect all this metadata and you collect the molecular signatures? At some point, all the experts around the world who are part of the HCA need to come with some sort of consensus. So defining really what that means, I think, is the next big challenge within a lot of the Atlas building projects. Is it a majority vote? Is there a way such that we could have a majority Atlas and then, much in the same way we have reference genomes and we have contigs that are sort of outside of the reference genome that people would download? Could we have this was the majority view and there's some minority sort of dissent? This is a big question which I'd like to know your opinion on. And as always, I'm interested in UI feedback. We have regular interviews with the various bionetworks. And this is the end. So lunch is coming soon. I'd like to thank the PIs who are part of this project. There's been David Osumi Sutherland, Davi, Fabian Theis, Niels Galenberg, John Marioni. The main PIs, originally it was Peter Karchenko, and the main PI of this project now is Chloe Villani in Boston. Special thank you to various people. I didn't have any time to talk about this, but there's more to CAP than just me discussing cell annotations. There's integrating these various cell annotation prediction algorithms within the web browser to make this accessible to the entire community. And there's many computational biologists with whom we work who are working on these in the Nir Yosef lab, in the Fabian Theis lab. So I'd like to thank John Erkenberg, Nir Yosef, Felix Fischer, and of course the HCA staff. We have Ellen Todres, Lucia Robson is over there, and many others, especially the bionetworks. They've been really great in the past year, giving us UI feedback. And of course, thank you to my team. There's Maxim Svetlakov, there's Maxim Solakov, Andrei, Roman, Mikhail, Denis, Nick, Lucene, Kirill, Kostya, Ur, Levan, Artyom, Pavel, Denis, Roman. There's a large team of people who have been working on this. You basically have front-end engineers, you have back-end engineers, making these UIs such that they work with the number of cells, and calculating differential expression doesn't take 20 minutes for you on your web browser. It takes a couple of seconds. Has been quite a lot of engineering work. So thank you to this team. They've been great. And that's the end. So.
Thanks very much, Evan. We're running behind, so unfortunately we'll skip the Q&A session. But if you have any questions, please approach Evan in the lunch break that we're going into right now. And we'll be back at 1:50 p.m., 1:50, after lunch for the second half of the cell atlasing session. Hope to see you all at 1:50.
Well, let me thank all the speakers and the chairpersons. Housekeeping announcements, like Shyam mentioned, that there are people who have joined online, so we have to sacrifice. Please do come back at 1:50. There are separate counters for non-vegetarian, vegetarian, and in the morning I said for vegan. There'll be a separate table, but they have put up a separate counter now at the end of the hallway. So please let's come back at 1:50. Thank you very much.